So, dear students, we are continuing with the endocrinology topic, and now we have to talk about invertebrate endocrine systems and vertebrate endocrine systems. So, if you talk about invertebrate endocrine systems, these are predominantly neurosecretory rather than classical. Neurosecretion, we talked about there are neurohormones and there are true hormones, or classical hormones. Neurohormones are those which are released from nervous system and then they enter the bloodstream to reach the target. But the classical are those which are released from a discrete structure which is called a gland or a cell or maybe a tissue, endocrine nature, and their secretion is released into bloodstream to reach the target tissue. So this is the difference. In invertebrates, as these are simpler organisms, neurosecretory systems are predominant there. In classical are few. In vertebrates, there are few neurosecretions and the more classical hormones. The second trend is increasing complexity in higher phyla. In higher phyla like Arthropoda, Mollusca, Echinodermata, the complexity increases basically. And of course, with that, number of and function of hormones, they also increase. In general, lower invertebrates like cnidarians, platyhelminthes, they have a limited number of neurohormones involved in their regulation of basically morphogenetic processes like development, growth, regeneration, gonadal maturation, and so. And other functions are, there are few functions that they do have. Few functions are controlled by many more means. In higher phyla, because there are more complex structures, there are more complex functions. So more complex endocrine control of functions is required. That's why they develop some more classical hormones like egg development and egg laying, like in Arthropoda, osmoregulation, heart rate maintenance, hemolymph, metabolic levels. What are the metabolic levels of metabolites in their hemolymph? That is their blood basically, and change in the colors and so on. Like in Annelida, mollusca, morphi, Arthropoda, like these animals have more developed circulatory systems and therefore have more effective circulation of hormones. See, this is very important, that if an animal is not having a proper true circulatory system, having hormones is actually difficult because hormone has to go to the blood to reach the target. So there should be circulation, not circulation. So circulation and hormone development is actually related. There is a considerable diversity in invertebrate endocrine structures and hormones in Arthropoda, mollusca, Annelida, and so on. So what are the generalized endocrine structures? We just give a few examples here. Like in insect reproductive system, there are testes and ovaries, and both are actually releasing hormones. Then there are also accessory glands and so on. So here you can see a male which is having testes, which is of course secreting a few glands, and ovaries in females which are also secreting a few glands. So this is one example. Then in mosquitoes, the Aedes mosquito, for example, the production of ova basically, it is important that it has to be strictly controlled, and this is mainly controlled by different hormones like and also releasing factors. Like here you can see there is an ovarian releasing factor after a blood meal which is released, and it effect upon EDNH, another hormone. This is released from corpora elata, the ovarian releasing factor. Its effect upon this juvenile hormone to EDNH, to follicle cells. EDNH actually causes follicle cell stimulation. Then ecdysteroids, ecdysone is a hormone which is there in the insects, in more insects. Here there are ecdysteroids. Then they affect upon the fat bodies, the target organs, and then in the fat bodies basically vitellogenesis, that is making the yolk is actually formed, because yolk is very important in an egg. So it activates vitellogenesis, and ultimately oocytes which are there, which have to uptake, that is take this yolk which is formed inside, so that the embryo can sustain on the yolk for some time. So ovarian releasing factor is one hormone, we can say. EDNH is another. Ecdysteroids, there are different types of steroid hormones which are there. Then ultimately it makes yolk. So making yolk involve many hormones. So there are many processes like that. Development of egg, we can say here. Then, for example, in their metabolism, many hormones are involved. Like you see, metabolism of fats involve adipokinetic hormone. Adipokinetic hormone, what it does, it affect upon an adenylyl cyclase in the cell membrane of the few cells. And what happened that they converts ATP into cyclic AMP. And cyclic AMP activates a protein kinase. It's a PK say protein kinase. Protein kinase converts inactive lipase into active lipase. And that active lipase break down triglycerides. Triglycerides are converted to diglycerides, which can go outside and can actually help in various processes. Just like trihalogen again affect upon adenylyl cyclase of another type, convert ATP to cyclic AMP, which convert another protein, which activates another protein kinase, and that protein kinase causes calcium intake. That calcium intake activates a PPK into inactive PPK into active PPK, and then that is phosphorylase kinase, PPA phosphorylase kinase, which converts glycogen into glucose 1-phosphate, and then another pathway ultimately is generated, which ultimately produces trehalose that goes to the hemolymph. So their metabolism is also controlled by a few hormones like AKH, adipokinetic hormone, and trihalogens, and so on. There are more like that. So these are the few examples. Like one of more we can quote, like the process of ecdysis. This is controlled by neurosecretions and hormonal secretions. There's the insect's brain is shown here, which is having neurosecretory cells which are making neurosecretions, which are stored in the corpus allatum and corpus caudicum, and both sides these are present. These are the glands, which produces ultimately juvenile hormone and PTTH, that is prothoracicotropic hormone. Now then there is prothoracicotropic, prothoracic gland is there. It affects prothoracic gland. Now prothoracic gland is a neurosecretion. Now prothoracic gland is a true gland which produces alpha ecdysone, which can affect the target tissues, which are the skin-like cells, and ultimately beta ecdysone is produced. Just like that, the endocrine hormone bursicon is also produced. So there are many hormones which are combination of neurosecretions here like and true hormones which are produced by glands. These are there in insects. So insects are more advanced forms of hormones. Here I'm leaving you with some examples. You can look at them by yourself. Hormone bursicon, its target tissue, tissue of origin. Then its structure, target group, what's the action, and how they are regulated. This is about bursicon, ecdysone, eclosion hormone, juvenile hormone, and prothoracicotropic hormone. Then here you can see spider, the common garden spider. Now what happened that these spiders also have different, generally there are 7 different types of glands which could be present in in the abdomen of a spider. Here you can see aggregate gland, flagelliform gland, so flagelliform gland, aggregate gland, cylindrical gland, and there are many more. Now these different glands actually produces different types of secretions. There you can see auxiliary special, some special kind of which have to make fiber like this. Then a drag line which they can produce is actually secretion of another gland. Basically they have to produce eggs. So when they are producing eggs, the tough outer silk of the egg is also produced by a gland. So there are many secretions which are produced by these spiders. So these are many invertebrate glands that we have talked about. Now we close this here, and then we'll continue with the vertebrate hormones in the next section. You can post your questions and anything on WhatsApp group or maybe in Moodle.